Hi class, in this recording we're going to focus on nervous system histology. So let's dive into it. First we're going to focus on cell types. There are two broad categories of cell types. We have neuron and neuroglial, or neuroglia cell. As we look at the neuron itself, this is the cell that will initiate and receive, or start and send and receive action potentials. So as we look at the generic parts of a neuron, this is our generic neuron. I want to emphasize that the cell body of the neuron is referred to as the neurosoma, and there's dark patches in the cytoplasm. Those dark patches within the cytoplasm of a neuron are referred to as Nissl bodies, sometimes also referred to as Nissl substance or chromatophilic substance in your lab manual. As we look at dendrites and axons for the cell body, dendrites are the smaller extensions that lead towards the cell body, or the neurosoma, and the axon is the singular long extension that leads away from the neurosoma. Now, as we're looking at the histology views, this view that I have right here is a very good histology view. It's more of a labeled diagram. So here we have the histology view. We'll start off with the neurosoma right here in the middle of our cell body, and then we have these dark patches within it, these dark patches within the cell body. So for instance, at the very tip of my mouse pointer right here, or the very tip of the mouse pointer right here, those are Nissl bodies from your lab objective sheet, or the chromatophilic substance. So those are the three things that I want you to be able to identify. We have a cell, we have substance or component of the cytoplasm, and then we have component of a cell as we look at these three different things. So make sure you pay attention to the wording of the questions on your lab exam. As we look at axons, I'm not going to ask you to differentiate the histology of an axon and a dendrite. From a histology perspective, it's just too hard. I don't like it. So as we're looking at this raw image right here, dendrites and axons are very difficult to differentiate from each other. So I'm not going to ask you to do that. More idealized diagrams like this that have the labels removed, those are fair game, though, for differentiating axons and dendrites. As we're looking at axons, though, I want you to be able to differentiate myelinated and unmyelinated axons from each other. So here is a digital histology slide that I have pulled up. For those of you curious, this is slide number 6 of peripheral nerves. And I like this image because it really does a good job of juxtaposing myelinated with unmyelinated axons. So let's start with the low-hanging fruit. Here are the myelinated axons, and we call these axons myelinated because you can see this white substance going around kind of the periwinkle or lavender axon. So in purple here we have the axon, and then the myelin goes around that axon. So those myelinated axons in this image are quite large. The unmyelinated axons are more difficult to identify. They are much smaller. So here's an unmyelinated axon. Here is another unmyelinated axon. These unmyelinated axons aren't going to have that very pronounced white band around them. Good rule of thumb on your lab exam is if I'm asking you what kind of axon is this, and I'm pointing to a region that kind of looks like hamburger, I want to hear I'm asking for an unmyelinated axon. And if the arrow is pointing to something that has a white band around it, instead of asking for an unmyelinated axon, I'm asking you to give me myelinated axon as your answer. So let's talk about the neuroglia cells. As we look at these cell types, we have astrocytes, oligodendrocytes, ependymal cells, and microglial cells. These four are located within the central nervous system. And then we have Schwann cells and satellite cells that are located within the peripheral nervous system. So let's start off with the CNS, central nervous system. While we're looking at the astrocytes, I want to emphasize that they have a star-shaped appearance, so they have kind of a starburst appearance, many small extensions coming from them. This is probably one of my more stereotypical images, I would say, of an astrocyte. So we have these They almost look like an asterisk or starburst, hence the root word astro. For oligodendrocytes, yes, this technically shows oligodendrocytes, but it's a terrible image of oligodendrocytes. It does not do a good job of illustrating what they look like. And this, again, doesn't do a good job. Also, we're not going to be using electron microscopy. I really want to emphasize with the oligodendrocytes that we have one cell body with multiple pseudopods coming off of it to myelinate multiple parts of multiple axons. So this image right here, this is an excellent oligodendrocyte image. And this is a fluorescent microscopy image. So the oligodendrocytes were chemically modified to fluoresce bright green when they were exposed to ultraviolet light or UV light. So right here, this fluorescent microscopy image, I love this image for oligodendrocytes. And then as we look at microglial cells, these can be thought of as the white blood cells or macrophages of the central nervous system. They oftentimes are going to resemble a small astrocyte in appearance. And then finally, we have ependymal cells. The ependymal cells themselves, I'm going to go, you know what, these arrows aren't doing a good job. I'm going to go ahead and outline them in green for us here. One individual ependymal cell is right here, being outlined in green. And I'll go back to the pointer mode, cursor mode, back to the main slide. So as we look at these ependymal cells, they kind of have a simple cuboidal arrangement, even though they aren't technically classified as epithelial cells. I also want to emphasize that they have cilia. So you can see those hair-like extensions. Those are the cilia of the ependymal cells. So those are our cell types, our neuroglia cells of the central nervous system. We also have some neuroglia of the peripheral nervous system. So as we're looking at these cells, we have just a standard peripheral nerve. I'm going to go ahead and zoom in on this just nerve right here. I'm just going to keep zooming in, and we can start to see axons. We see a lot of myelinated axons. So like right here at the tip of my cursor, there's a myelinated axon. And then you can see that there's some white myelin, a ring of white myelin going around the myelinated axon. That myelin is a substance produced by Schwann cells. So instead of looking at a cross-sectional view like I did up here, if we look at a longitudinally sliced view, we can better see the Schwann cells. So while we're looking here, we can see these white masses. Those white masses represent the Schwann cells. And here's another Schwann cell right here at the tip of my mouse. And as we're looking at these Schwann cells, where two Schwann cells merge together, right here, for instance, we have a myelin sheath gap or Neuron VA. So there's a lot of things I can ask you at this. I could ask you, again, what kind of axon is this? It's a myelinated axon. I could ask you, what's the substance? The substance is myelin. I could ask you what the cell type is, and that is a Schwann cell. Or I could even ask you what component of a neuron is present here. Actually, I won't because I don't have the asterisk next to axon. But if I asked you that, I would want to hear axon. But to be true to my word, axon itself does not have an asterisk. So I would either ask you for a myelinated axon or unmyelinated axon on your lab exam. Next, we have satellite cells within the peripheral nervous system. These satellite cells are going to be visible when we look at the ganglion within the peripheral nervous system. So I'm pulling that up right now. And I'm not a big fan of that image. The longer I do this, the pickier I get. So I'm going to go ahead and first give us some context. Let's look at the dorsal root ganglion right here. So this is from a spinal. Actually, let's really give you some context. So here's the spinal cord cross section in all of its gory glory. And then right over here, we have the dorsal root ganglion. Let's go ahead and zoom in on that region right here. And if I keep zooming in on that dorsal root ganglion of the spinal cord, I can see large neurosoma, large cell bodies of neurons. So I'll go ahead and circle a cell body of a neuron right here. There's that cell body of the neuron circled in green. And then around the perimeter of the cell body of that neuron, we have satellite cells. And those satellite cells, let me go ahead and clear the screen. Those satellite cells are going to be these small cells. So right there's a nuclei, there's a nuclei, right here's a nuclei, there's a nuclei. These are the cells that wrap around the outer perimeter, around that neurosoma. So if we go back to, let me clear the screen again here, that initial image I had pulled up, we can see the satellite cell nuclei very clearly in this image. This image is really optimized to highlight satellite cell nuclei. So those are the two neurosoma, or excuse me, the two glial cells of the peripheral nervous system, and then the 4 glial cells of the central nervous system. And then finally we have connective tissue wrappings of a nerve. A nerve is a bundle of bundles, kind of like a muscle is a bundle of bundles. So instead of having an epimysium, a perimysium, and an endomysium, we have an epineurinium, a perineurinium, and an endoneurinium. We have the same nomenclature scheme. So while we're looking at our parts of a nerve, first, this is just a cross-sectional view of a new nerve that's zoomed way out. We'll start on the surface or from the outside and work our way in. First, we have the epineurinium. This epineurinium is this thick connective tissue sheath wrapping that is on the outer edge of the nerve, and sometimes will separate major nerves from each other that are bundled together. And then within the nerve, we have fascicles, which are bundles of axons, and lining individual fascicles, we have a perineurinium. So I'm actually not a big fan of the fact that this was highlighted right here, because from what you can see as a student, it's not very clear how those are different from each other. So I'm going to go back to our lab objective sheet, just because I have the hyperlink available. So as we look at our epineurinium, the epineurinium is going to be this dense, irregular connective tissue around the outer edge. And then the perineurinium is going to be within between the fascicles. So as a good rule of thumb to differentiate them from each other, we have a big fascicle right here. We have another fascicle right there. And then separating the fascicles, I'm tracing it in green, we have the perineurinium. So what I like to do to make things extra clear on the lab exams, I typically like to point to a perineurinium that is right in between fascicles, or if I go for a most superficial view or most outer view, like I have in this top arrow, that would be a epineurinium. The endoneurinium is more difficult to find. That's going to be wrapped around individual nerve fibers. So to find the endoneurinium, you've got to zoom in close. The endoneurinium is the pink band around the myelin sheath gap that goes around the axon. So we have a bunch of myelinated axons in this view. Wrapped around the myelin is the endoneurinium. Let's go ahead and pull up an endoneurinium view here within digital histology. As we're looking at this view right here, we can see the endoneurinium is that dark pink band. I'm also liking the perineurinium in this view, that dark pink stained bundle of, or cluster of connective tissues. I'm going to advance just a wee bit here, just so you can get another view of the endoneurinium. In this particular slide, the endoneurinium has been stained light green. This is a pretty special slide, and it's unfortunately I don't have any like this for my in-person teaching experiences. Anyways, back to our lab objective sheet. Those are all the histology objectives that you need to be able to identify for your lab exam. If you have any questions or comments, please post them in the class discussion boards or feel free to shoot me an email. And as always, happy studies.